Okay, so to work in your lab report, so again, I'm going to do a short video for some of you, and you can stay longer in case that you have a doubt. So key information that you need to know, that in your lab manual you will find this, and in the next slide you will find all the different indications or the guidance that you need to know what kind of answer you need to go and write in your textbook. Important, I here, if you have any doubt, I post an example of the calculations that you are supposed to do. They are imaginary calculations with an OR set of data, and I made an Excel table in case that you wanted to do it. You can also do it manually if you find that Excel, you don't work with Excel. You can also, like, or with other software, or with the, yeah, this chi-square, you know how to do it. And I also have a Word document that you can download where I create all the tables that you have to fill, where you have where you can add all these things, when you can have all the formulas written, so it's much easier for you, and you just have to write the analysis and the correct information. So in this way, you can finish the lab manual and faster, and do it in the right way. In your lab manual, you will find these indications or this guidance for your lab report. And I'm just going to, through this video, to try to explain some of those things. If you want, you can watch it, or if not, if you don't want to watch it, it's fine. The idea that I will go through every single steps and give you an example of how to do it with a previous with an imaginary data. So if you have any doubt, you can go through this video and say, okay, I have a doubt about this, if I did it right, so you can double check. First thing that I have to tell you is that here the title of the lab, you're just repeating some things. So it can be something like this. There is no need to, I'm not going to grade the title. Yeah. The introduction, there is no need for introduction. And you can go directly to answer all these questions, and all the things that, these are the things that I'm going to grade, and that you get the right information here. So remember, I'm not going to grade the title. Don't think a lot about it. It could be this one. There will be no introduction, and you can go right away to try to answer the questions. Okay, so first question, here it is. So it says, report the expected genotypes and phenotypic ratio in the F2 of each cross. Okay, so you remember this. So here this will be the expected data for your chi-square. Okay, and here what I'm looking is, you have here the parentals. You don't have to make a Punnett square for the F1. You already did it before, so I just want that you do that. That you write here the two types of genotypes that you get here, and here I want that you do the F2 of the expected ratio. Why? Because this will be important to compare your to make your chi-square, and this can give me information if you made the correct assumptions, and I will be able to grade it. Okay, remember, you already submit this, and I am supposed to grade it before, so if you have any doubt, let me know. Okay, so remember, where are you going to get the expected data for your chi-square? From here. Okay, this will be your expected data. Okay, so you already answered question one. It was pretty easy. The question number two is use these tables, the tables, to test a hypothesis. So that white and sepia alleles are both recessive on different chromosomes that are, and white is being located in the X chromosome. Yeah. So how you do that? You have to show a full chi-square analysis using the whole data from both reciprocal crosses. So here in this video, I'm just going to show you an example of what you are supposed to have in the observed data and expected data. And this is just an example in case that you have a doubt, as I said before. Okay, so to answer the question that of question number two, you need to know very well the chi-square. So you need to know how to use this formula, and if you have any doubt, remember there's a video lecture, and remember the textbook goes and talk in detail about this. So please go and review these topics so you can answer properly this question. And I know that you know that you should analyze a ratio your expected data. The observed data is here, and as I said before, I will send you this information by email or I will post it on Moodle for you. Okay, and these are the type of information that you will need to solve this chi-square. So remember something that we cover in the class and is in your textbook, that there are always in a statistical test a null hypothesis and an alternative hypothesis. In this case, I'm going to show you just an example of the observed data, and this is an example of your expected data. So what we are trying to show here with the chi-square is how similar our what we observe to what we expect. Yeah, so we said that we should have these ratios. Yeah, we shouldn't have here, for example, a female in white in this cross, and we are supposed to have a probability of 4 out of 16 to get males for this white gene, yeah, in this cross. So here, for example, we get 0, so that is great. And we have here 103, so we need to see if 103 is around 4 out of 16 of 461. So I will show you in a second how to verify that, okay? But in other words, what we are doing with the null hypothesis is we are comparing these two tables, yeah, all the cell values, and we're saying, you know, these are very similar, yeah, and maybe here we were expecting 100, for example, but we got 103. Why? Because there is only a difference to random fluctuations that are very small. Yeah, maybe we should have got 2,000 flies, 5,000 flies, 10,000 flies to start getting closer and closer. Or maybe, you know, it was just there is all these variations, so maybe there is only random chances. Yeah, small changes that are not significant. Okay, and so conclusion, null hypothesis is that what we observe is exactly what we expect, and the alternative hypothesis is the opposite. Is that here, for example, imagine that we get here instead of um, we were expecting females. Here we were expecting 0, and here we get 1,000. So we said, okay, here this value is much higher. It's very different from 0. So maybe the difference is not only due to a random factor, but there is also another factor that is significant and is very important. So this is something that we have to consider. And in this case, we say that that we reject the null hypothesis. Yeah. So in the first case, in this one, we say we don't reject the null hypothesis. In this one, we say that we reject the null hypothesis because this one will be the table. Definitely, this is not what we're expecting. In other words, in this case, maybe there was something wrong in this experiment. Maybe uh, there is not enough numbers. Maybe the way the way that we were expecting the mechanism of inheritance is different to what we expected. They could be this is what we are supposed to write in the analysis. Should we repeat the experiment, or there is definitely something wrong in the way that the mechanism that we were proposing? Okay, so now let's go to the next slide. Okay, so now that that is clear, that you know all the information about the chi-square, that you review this very quickly. Now let's try to use this example of the data for the cross A and here for the cross B. Let's try to start trying to do the chi-square. Okay, this step is very easy, and is that here with the observed data you have actually numbers of flies. Yeah, in the beginning and in the expected data, as you took this from a chi-square, sorry, from a Punnett square, you just have probabilities. Yeah. So now you need to turn these probabilities into number. Yeah. So this will not be. So for example, wild types, females that you have here is 6 out of 16, while the males is 3 out of 16. And these are probabilities. Now you need to turn that into numbers. Okay, so let's do it. It's very easy. So here I have in a simple way how you do it. So for example, for the wild type, you have 261, yeah, for males and females together. So what you have to do here is just to take the total number of flies here, and multiply it by by the probability. So here it will be the probability 9 out of 16 multiplied by the total number of flies, that is 461, and here you get your expected data, that is 259.3. Okay, now let's try to do the same thing with the sepia. Okay, so the sepia will be this one. Yeah, this is the one in brown, and it will be here, so you don't get confused. Okay, so while you do it, I will delete this to make it easier for you. Try to get the correct data. Okay, so let's do it in this is the closest to brown that I have here. So we have the sepia, so you have 97, perfect 97 here. Now let's go and get the expected that is here. So we have the probability is 3 out of 16. And if you look, here is the probability 3 out of 16 from a total, and you multiply by a total number of flies. Here you have the total number of flies. So you get 86.44. So you were expected to get 97. Now you got 86.44. So with the chi-square, we will see if this is good enough or they were very different. And now let's do with the white one. So now you can start working in the white one. So here, white one will be this one. Observe 103 in total. Yeah, now let's go and look for the expected. So we have 4 out of 16. Here in the total, all that we are expecting is 4 out of 16. Here it is. And now you multiply by the total number of flies, and it's here. 461. So you were expecting 103. Now you got 115.3 flies. Okay, so it's pretty easy. And if you have any question, let me know. So what I did in the previous slide is take this observed data, all these totals, and in this table you will see again the new expected data, the adjusted data that it will go here. As I was working in Excel, it was pretty easy for me to also get the data from all the other places. So for example, here I still get, for white females, I still get a 0. And for wild type males, I also got 172. And I was expected, I was expected to observe 172. And I can see that I was expected that I got 183. Okay, so this is just, although we're going to calculate the chi-square only with these values, this information can be useful if I want to make chi-square not with totals, but with the females, yeah, by gender specifically. Okay, so let's continue. So remember, you have to show your work for the full chi-square. So I already, if you want, in the Word document that I already gave you, in addition to make this table with these 2 columns, I also made one where you can add all this information. And if you like Excel, I already create also an example of the table that will help you to do all the calculations. Here, make sure that most of the numbers adjust. Sometimes, the Excel allow many more columns, many more decimals. but the idea is not that it's perfect, that is going to be that I'm not going to grade that you have exactly the same numbers, but you should have to the same conclusions and very close numbers. Yeah, they don't have to be exactly the same, but if you have instead of 2.6 you get 6.9, then definitely they should be a problem. Okay, so remember, check if you want. The Word document is there, as I said before, and the Excel is there if you know how to use Excel, or you can do it by hand and with your calculator. Okay, for your analysis, remember you will need the chi-square that you already got it from the previous table. You need a critical value that you will get it from this table, but before of using this, you need to have the degrees of freedom. How do you get the degrees of freedom? You need to, if you remember the video lecture, you will need to count how many treatments you have. So you have 1 that is wild type, 2 that is sepia, and 3 that is white. So it will be 3 minus 1, that is the formula, groups minus 1, so it will be 2. Now the degrees of freedom, it will be here, it will be 2. And the probability that we want is, in this case, 0.05, okay? So what you get here is 5.991. And if you see here, our chi-square is much smaller than the critical value. So because the chi-square value is less than the critical value, we don't reject our predicted ratios. In other words, the two tables, each one of those cells seems to be similar, yeah, enough. So we don't reject our null hypothesis in this case. We don't reject our null hypothesis. We only will reject it if it was going to be higher here. If we have 7, 8, 20, yeah, in this case we reject, yeah. Here in our case, as is lower, here is we do not uh, reject. Sorry, not doesn't look very good when I write in using the mouse, but here you can see all the analysis if you want to read it, and where you can find it in the textbook. Okay. Um. Okay. So you not only have to do this for one cross, but you also need, so for the cross A, but you also need to do it for the cross B. Do not make this video too long. I just, if you want to see the calculation of this example, I have it in the Excel table, and you just have to, for your lab report, you just have to fill again all these things, and you don't have to hand in an Excel table. That Excel table, an Excel file. I just want that you just have it there in case that you have doubt with your calculations. Okay, so remember, if you have doubt with the cross B, I already create an example there. I'm not going to record it. You can view if you have any doubt the results. So we already solved the question 1, the question 2. Yeah, we have done a chi-square. We came to some conclusions. Now in the question number 3, they are asking you, okay, what about if you have a possibility that, and some of you already asked me this, when you have a homozygous recessive for both white and sepia, the double mutant, what happened is that you are always analyze the adults, yeah? But what about if they died in the larval stage, yeah? So they are not alive anymore. So we never see them, yeah? Yeah, so we get these phenotypes die during the embryonic stages, and where we are counting is actually the white ones. Yeah, we are not counting actually the real flies. So so let's test this hypothesis, and let's do it together. How do you have to modify your chi-square tables in case that if you want to test that hypothesis. So let me again show you an example of how you are supposed to do it, and then you can do it with your data. So to test this hypothesis, in the section 3a, they said for one of the reciprocal crosses, and it's the one that you choose, yeah, is your choice, you need to check this hypothesis when you have 100% lethal flies prior adulthood. So how do you do it? You need to go again to this table, and you need to look for the double mutants. So here I put the males, but you can also look for the females that are double mutants. And you remember before you used to have a ratio of three phenotypes: white, sepia, and wild types. Now you're going to have 4 ones. And my suggestion is that remember the 9331. So here you have 4, but I don't think that this will look like 9331. It will look very different from that one, like the previous one. But take into account you need to add this new group, that is the double mutant. Okay, so let's continue. Let me show you the example. Okay, so here, as I said before, now this is the 3A. In the 3A, you have to choose one reciprocal cross and the cross that you want, the A of your or B, and you have to use the chi-square to test this hypothesis that 100% of them that they are double mutants, they died before adulthood. So from your observed data, it will look like this. This will be the same, and this one you will have an extra row that it will be double mutants. Okay, so easy. The numbers will be exactly the same, just that here all of them will be 0 because your hypothesis is that all of them died. Okay, so and your expected data that you had before is here. Now your expected data, it will be different because you will have these guys. Okay, so now I want that you see compare the expected data before and the new expected data with the double mutants. While I delete this, all this information, do you see any difference in the tables? There are two main differences, and I want that you think about what are the two main differences and why. Okay, so I'm going to use something very colorful. Okay, purple. So first difference, I know you saw it, is here. Here and here. So there is this different. Okay, and so before you have 4 and now you have 3. So why? Because one of the white males, it was actually in the table A, it was a double mutant. So now where it goes? It goes to here. Yeah, to the double mutant. Let me draw it better. To here. So we remove one of the 4s and now it turned into a 3. Okay, so this is how you change it. And now there is another difference, and the other difference, I'm going to label it here, is the 15 in red, and here you used to have 16. So why you have the probabilities? Now we have 15 and 16, and here if you see here, instead of you have a 7 and here you have an 8. So why we have those differences? Okay, so think I'm going to delete this too. So the difference is because before you add all these values 9, 4, 3, and you get 16. Now you have 9, 3, 3, and this one that is supposed to be the extra value, you don't count it because the fly died during embryonic stages, so you cannot see it. So you suppose that is 0 because you cannot count it. So you, instead of getting 16, you get 15 because you miss one, and here you miss the other one. So there's one that is dead and you don't count it. Here you can write like one that is dead in here. Either way, the idea is that you cannot count it. Okay, so this could be 0 and this would be 0. Okay, so that will be very important for the calculation, and I will show you that in a second. Okay, so this is pretty easy. In both of those tables I have cross A. Here in the first one I have it for the question number 2. Let me write it with blue, that is easier to see. And this one is for cross 3. Here all of them, these are from adults that are viables, all of them. And here is for the ones that are in which the double mutants are lethal when they are adults. Okay, so as I said before, there are two types of changes. One change will be that all of them will be, instead of 16, they will be 15. And the other change that you can see here is that in the white, instead of having 4, this you will have 3. Yeah, so this will create a lot of changes. So while before you had 259, now you're going to get 276. Why? Because you divide, instead of 9, divided 15 instead of 16, and you still multiply by 461, so now you get this number. For the white one, you have more changes. Now you get 92, as is shown here. Yeah, before you used to have 115. Why? If you go to the previous table, it was 4 out of 16. Now it's 3 out of 15, so you get a completely different value. And here in sepia, you also get a different value because you have 3 out of 15. Yeah, so here before it was 86, now you get 92. Look, now white and sepia, the expected values are the same. Why? Because now you're divided both by 3. And here you expect 0. Okay, I know that I went a little bit quick through this explanation, but I didn't want it to be so repetitive. If you have a question, you can go and watch the previous slide or the other source of information, or you can just send me an email and check those things. Now let's go to the analysis. So now, to do your analysis, you have to first have the table with the chi-square. So now, remember, observed data will be the same, while the expected data will be very different, as I already explained. And remember, this is for cross A. For cross B, this number can change again, and also the other white can also change again. This one here, this can also change, so be careful with that. And now you get all this information, and you got 239 when you add all these values. Okay, so pretty easy. Now let's go and see if this value is we accept, no, we reject the null hypothesis, or we don't reject the null hypothesis. So to analyze the information is pretty easy. First thing that you have to do is to calculate the degrees of freedom. In this case we have 4 minus 1. 4 is the numbers of groups, so you get 3. And if you have a doubt why 4 groups, is because you have, in addition to wild type, sepia, and white, now you have an additional group that it will be the double mutants that are lethal. Yeah, although they represent 0 in most of the calculation, they are a new group. So now you're going to get this degrees of freedom that it will be 7.8. So here it will be, and here is our chi-square. So again, this area here is we do not reject, and here is we, and let's put it in blue, and here is we reject the null hypothesis. And so our data is not here, our data is here. So conclusion, we don't reject. Yeah, so pretty easy. And now to finish, So now I want that you write for 3B a concluding paragraph. Yeah, I want that you don't need to write the history of genetics and write 20 pages. I just want that you write a concluding paragraph. And for example, here I want that based on your chi-square results, you tell me, in question 2, I don't reject the null hypothesis. Yeah, and in question 3, for example, for the thing that we did, we didn't reject it either. So based on that, what you can say? You can talk about the numbers, yeah, say like maybe we need more numbers because maybe you can accept this one and the other thing is reject this one, and this will make more sense than accepting both. So how do you explain that? And there is any way that you can test your conclusion based on the genetics test that you have done, all the things that you have analyzed, that you have learned in this class? Is there any other additional experiments that you can do that it can help us to solve why, for example, we got this inconclusive data? Yeah. So your data can be different, but you have to write this type of analysis. Again, if you have any question, just let me know. Have a good day and bye.